understood how exactly a peptide bond is formed, let us look at other structures that are formed in a protein. We know that with the bond angle that can vary dramatically or tremendously, there can be several conformations possible in a peptide bond. Now, which conformation is stable? That depends on the hydrogen bond formed between the carbonyl carbon and the NH group. So that determines which is the most stable form. So let us talk about two good possibilities. One possibility is hydrogen bonds can be formed from one polypeptide chain with the next polypeptide chain. So therefore, we say that it is an interchain hydrogen bond formation. So with this interchain hydrogen bond formation, it will allow a peptide bond to have a very relaxed bond angle. So this kind of a structure is formed in beta pleated structure. The next possibility is if the hydrogen bond can be formed within the structure, intrachain, that leads to formation of alpha helical structure. Let us try to understand how exactly an alpha helical structure is formed. Assume you are driving your car to the top of a mountain. So, at the top of a mountain, assume this is the mountain, you can go in two ways. One possible way, you can go in this direction. You start your journey from here, you go from here, you go from here, and you go from here, and you reach the top of the mountain. That is one possibility. Okay, you will take very sharp bends. So these are sharp bends that will take you to the top of the mountain. Another possibility is, let us rub this. If you do not want to take sharp bends, you go around the mountain. Okay, how do you go around the mountain? From here, you will go here, and then you will go around the mountain. Okay, you come around the mountain, then you will take second round, go around the mountain, you take the third round, go around the mountain, and you reach on top of the mountain. That is the next possibility. Go around the mountain. If the journey may be longer to reach on the top of the mountain, but you reach on the top of the mountain without taking sharp bends. Okay, suppose we remove this mountain, we remove the mountain. Okay, we remove the mountain from only the roads are existing. This is how an alpha helical structure is formed. Now remember, it will not be narrow like this, it will have the same dimension, same diameter. So this gives you an idea about how an alpha helix is formed. Now remember, this is moving in this direction. So therefore, this is right-handed helix. And if you start viewing from here, it is going away from you. So therefore, this is the N terminus where you began your journey, and this is the C terminus where you will end your journey. It is moving away from you. The rotation is moving away from you. This is how we understand a right-handed helix. Max Perutz, one who studied the structure of protein, he gives a beautiful analogy. And the beautiful analogy is an alpha helix structure can be considered to a spiral staircase. So a spiral staircase, you have one step, then the next step is here, then the third step is here, okay, sorry, not here, here, and the fourth step may be facing you, and then the fifth step. So this is how a spiral staircase is arranged. Now each of these steps are considered to be an amino acid. Okay, one amino acid is arranged here, This is the second amino acid. This is the third amino acid. The fourth you are not able to see because it is facing you. And this is another amino acid. So you can arrange amino acid in a spiral form. Okay. So assume in the distance between two amino acid. Distance between two amino acid is 1.5 Armstrong. Distance between two amino acid is 1.5 Armstrong. So therefore, you can find out in one ring structure, okay, one ring structure, what will be the thickness in one ring structure. You will be able to calculate that. Let us understand in the basic structure. So I am going to rub this portion after having understood how exactly the spiral is formed. Let us assume we have one amino acid connecting with this is amino acid number 1, amino acid number 2, amino acid number 3, amino acid number 4. We want to make a connection, alpha helical structure between amino acid number 1 and 2, between 2 and 3, 3 and 4. You will get this kind of a zigzag structure. Like you are climbing the mountain, not traveling around the mountain, but making zigzag structures. So what will happen is, this will not give us a helical structure. We say that n is equal to 2. This will not give us a helical structure. It will give us a flat ribbon kind of a structure. Let us make an alpha helical structure with 3 amino acids. So therefore you will get a structure like this. So in the first amino acid, 3 amino acids must make. So therefore the second one is the third one. So that is making one cycle. So therefore we say that n is equal to 3. You can also make n is equal to 4. So it will have a structure like this. So first amino acid, second one, third one and the fourth one. That is making one ring structure. So this is n is equal to 4. What is the difference between n is equal to 3 and n is equal to 4? It is in the diameter. In the case of when n is equal to 4, the diameter will be broader. It will be larger. And it will be all the more larger when n is equal to 5. Because 5 amino acids you need to incorporate in one ring structure. You have a very broad ring. What is seen in normal protein alpha helical structure is n is equal to 3.6. N is equal to 3.6 amino acids makes one ring structure. And this is very interesting. Now, having understood the structure, how peptide bonds are formed, how this 3.6 is formed? 0.3 amino acids of one amino acid is the second amino acid, is the is the first amino acid, is the second amino acid, is the third amino acid, and 0.3 of another amino acid. That makes 3.6 amino acid will make one round, one cycle, one ring. So we have seen that distance between each amino acid is 1.5 Armstrong. So you multiply by 1.5 Armstrong, that will give us a value of 5.4 Armstrong. So that height, so therefore from here to here, this is the height, distance between two rings. This is what we call it P. The pitch is equal to 5.4 Armstrong. This is a very important value, because based on this value, we will look at some of the problems that can be addressed. If you are given the total length, how do we find how many amino acids are there? So therefore, another interesting aspect is we have seen that 3.6 amino acids makes one ring, which means 7.2 amino acids will make 2 rings. If you want a whole number, that means 4 amino acids, this is 14.4, Add one more, that is 18. You get a whole number. So how many ring structures are required? 5 ring structures will make a whole number. You will see that this nitrogen, and after the 5th ring, that nitrogen is exactly parallel. So hydrogen bonds can be established between this helical structure. So this is how an alpha helical structure is formed. So therefore, what is existing in our body is between 3.6 amino acids. So this is called 3.6 helix. If you count the number of atoms, it is roughly about 13. So therefore, it is known as 3.613 helix. Okay, it is called 3.6 helix. Normally, an alpha helical structure forms with about 12 residues. That is normally found in protein. But there are reports it can have maybe 50 plus amino acid forming, a 53 amino acid forming a helical structure are also reported. You will find alpha helical structure very prominent in transmembrane protein. Even on the 7 transmembrane receptor is a collection of alpha helical structure, about 7 alpha helical structure. Leucine zipper, we will be looking at leucine zipper. It is a collection, it is an alpha helical structure which forms part of the protein. When we discuss about alpha helix, we should make a reference about pi helix. So a pi helix, written like this, pi helix. Pi helix is also known as alpha bulge. So it bulges out. How exactly a pi helix is formed? So we have seen that there are, the linkage is either n plus 4. So that is the 3.6 amino acid, it is linking and it is forming a ring structure. When it is n plus 5, what will happen? The diameter changes. So you can see that here I am adding, so therefore this portion will bulge out. This is the normal diameter and one ring will be slightly larger. It will bulge out. Because of this bulging out, this is known as either pi helix or alpha bulge. It is also known as alpha aneurysm. A-N-E-U-R-I-S-M, alpha aneurysm. So you will see that it is in this helix, it is 4.4 residues are forming in the helical structure, which means it has got about 16 atoms. So therefore it is 4.416 helix also. Now, how exactly this is formed? So this is formed because occasionally a new amino acid is introduced. How can new amino acid introduce? Maybe some mutation happening in the structure. So therefore this is seen only occasionally, maybe in one ring, at the most the second ring, not beyond that. So it has a very important aspect also. So these kind of pi helixes are observed in order to find, make them as a marker for a particular protein. So they may have a particular function to perform in a cell, very specific functions. You see how variations can be brought in with the help of pi helix in the structure of protein and its functions. Now let us look at the microenvironment of an alpha helical structure. We know that now near the amino terminus there will be a positive charge and near the carboxyl end there will be a negative charge, and this is how the polarity is shown in the alpha helical structure. Now this alpha helical structure has to be stabilized. Let us see how this structure is stabilized. Which are normally the amino acids found at the N terminus. So you will find amino acids like threonine, serine, aspartate and asparagine. So these are normally found amino acid near the N terminus. Now these amino acid, they will interact, they will form a hydrogen bond with the amino terminus and they will try to neutralize the charge. So therefore we say that this forms something called an N cap. Okay, and a cap is put in order to stabilize the N terminus portion of the alpha helical structure. What about the C terminus? If the C terminus will have, it has a negative charge. So therefore you will find amino acids like arginine, lysine, etc. present at the C terminus. So it will stabilize, it will neutralize the charge and it will stabilize the structure of an alpha helix. You know that near the C terminus you cannot have an acidic amino acid like, let's say, aspartic acid. So if it is an aspartic acid, the pKa value will be too large, too high. So therefore this positively charged amino acids are introduced at the C terminus in order to stabilize, establish hydrogen bonds and stabilize the alpha helical structure. So this is how the alpha helical structure is stabilized by the microenvironment of amino acids. There are several aspects one has to understand about the alpha helical structure. For example, amino acids like glycine, lysine, arginine, etc. are not present in the alpha helical structure because they can cause, the R group of these amino acids can cause repulsion. So a repulsion, you know that it will destabilize an alpha helical structure. Similarly, valine, threonine, isoleucine, they all have a beta carbon atom and there is a branching. Because of that, it will cause a steric hindrance in the alpha helical structure. So these amino acids are also not found in the alpha helical structure. 3 amino acids, that is serine, aspartic acid and asparagine. If these 3 amino acids are present in the alpha helical structure, what they will do, they will compete to have a hydrogen bond with the carbonyl carbon and with the amino nitrogen. So therefore these 3 amino acids are also not normally present in the alpha helical structure. Glycine and proline are also not normally found in alpha helical structure because glycine is a good hydrogen acceptor and proline is a good hydrogen donor. So therefore they will enter into hydrogen bonding and it can destabilize the alpha helical structure. Another important aspect is, you have seen that an alpha helical structure is made up of only L amino acids. D amino acids are not normally present in any protein. Even if D amino acids are present, they are limited to bacterial cell wall or certain antibiotics. So otherwise, all proteins are made up of L amino acid. This has some evolutionary aspects as well. So if at all a L amino acid has to be converted to a D amino acid, it has to be a enzyme based mechanism. Then only L amino acid can be converted to D amino acid. Occasionally in here and there may be a D amino acid structure existing transitorily for a while. Otherwise, normally a protein structure is purely made of L amino acids. So these are the points that we need to remember about the nuances of alpha helical structure. This discussion on alpha helix will not be complete unless we say a few lines about the role of proline in alpha helical structure. Proline is not normally found in alpha helical structure. So we have seen earlier that proline is, first of all, it is not an amino acid, it is called an amino acid because proline has no NH2. It has no NH2, it has an NH group. So therefore, it will create a kink in the peptide bond, in the structure of alpha helical structure. So it creates a bend. We have seen that it is promoting a cis configuration. Because of this structure, proline is a destabilizing amino acid in the alpha helix. And also understand that proline, when you look at the carbonyl carbon of proline, that is more negative, because if the ring structure pushes a lot of electrons to the carbonyl carbon atom. So therefore that becomes heavily loaded with, there is a larger density of electrons at the carbonyl carbon. And proline is highly soluble in water. So it interacts with the water. It forms hydrogen bonds with water. Highly soluble. Therefore you can find, if at all there is proline present in alpha helical structure, you will find them close to the N-terminus portion of the alpha helix. And it has an advantage. Since it protrudes, it is not allowing a real ring formation. So it will form like, it will act like a linker. So if you look at the structure of a proline, this linkers becomes very important. Take the structure of an enzyme known as pyruvate dehydrogenase. You know that pyruvate dehydrogenase, the different domains are connected by linkers. Now these linkers can be an assembly of proline and glycine. So a polyproline structure, which is very important, and we need to look at how exactly this polyproline structure is formed. So it is a PXXP structure, proline and any other two amino acids. Now the polyproline structure, it cannot have, in the ring structure, it cannot have the 3.6 amino acid ring structure. It will form with the 3 amino acid ring structure. What happens with the 3.6 amino acid? If the bond angle becomes about 100 degrees. Whereas in the case of PXXP, if the bond angle is 120. So therefore only 3 amino acids are allowed. Let us understand how a polyproline structure is formed. So we have N, so that is the first amino acid. Now connected to the next amino acid. Now that is connected to a proline peptide bond. Now proline linked to another amino acid. So it is PXXP. So therefore if the next amino acid is linked, and therefore if the next proline is formed, so this is linked to another amino acid, next amino acid, next proline, and this is how the polyproline helix will be formed. Only 3 amino acids, 1, 2, 3, only 3 amino acids per turn. This is the interesting aspect of a polyproline structure. So such structures are also found in the alpha helical region. Now these structures can act as a kind of a linker. It will give greater flexibility to between domains, and it is able to move. Polyglycine is also found in the alpha helical structure. So polyglycine and polyproline are rarely found within the alpha helical structure. This is the point that has to be noted. So therefore, the kind of polyproline ring structure we have seen is known as polyproline type 2. Polyproline type 2 ring structures. Polyproline type 1 ring structure is, it's a theoretical ring structure that will have more of cis bond. So it is a theoretical ring structure. Now, when we speak about polyglycine ring structure, see, glycine is a typical amino acid. If the R is equal to H in the case of glycine. So strictly speaking, we cannot speak about either right-handed helix or the left-handed helix in the case of a polyglycine. But all these polyglycine, polyproline, etc. are important to give greater flexibility to a protein structure, and that is the biological significance of polyproline or a polyglycine structure in a protein. So what we have seen in this session, we have looked at how exactly alpha helical structure is formed, what contributes towards the structure that we know today. how it is exactly formed, what all stabilize that structure, how the microenvironment will stabilize the structure, and what are the other nuances like polyproline or polyglycine structure which is seen in alpha helical structure. So these are the points that we have seen in this particular session.